Thank you so much. It's my pleasure to be able to speak to you all today about some wonderful life extending lab tests. All of you, I know that you are incorporating a variety of strategies in order to increase your longevity. And when I was asked to talk to you all, I said, you know what? Let's talk about different lab testing. How do you know if the strategies that you are incorporating, if they are working? And so hopefully I'll be able to share a few new insights that will be able to assist you on your journey. So we'll start off with just talking about a few, and this is, just 5 of many mechanisms of aging that we would want to address. So the first is oxidative stress, and just to give you an overview of the flow and the direction that we will be going in today, we will talk about, have a couple of intro slides on these different mechanisms of aging. And thereafter, we will talk about the different testing ways to measure those mechanisms of aging. So we have oxidative stress, glycation, methylation or insufficient methylation, mitochondrial dysfunction, and chronic inflammation. So let's just jump right in there. Oxidative stress. At Life Extension, we have this tagline in all of our new hire classes. We have wellness specialists that assist customers with questions over the phones, our doctors. We show this slide. It's the buildup of free radical damage over time. We know that we are going to be exposed to free radicals. They are everywhere. You're breathing oxygen. Hey, that's a free radical. Your body's production of energy, and we'll talk about energy production in the mitochondria, but one of the negative implications of creating energy, creating ATP in the mitochondria, is the production of free radicals, hydrogen peroxide, and a host of other free radicals. Now, our body needs to be able to combat these free radicals, and naturally, we have an internal antioxidant system. Many of you are probably taking glutathione. That's a natural internal antioxidant. Catalase is another one that you may hear of. These are natural internal antioxidants designed to target free radicals that we encounter every day. So what you see on this slide, this is just showing a healthy, stable molecule. You can see they're paired electrons along that stable molecule, and then you see an unpaired stable molecule. Let me make sure you can see my pointer. So this is an unstable molecule, and that's where we have the X there. And then you have the antioxidants, which I'm sure many of you are taking. Vitamin C is a wonderful antioxidant. PQQ is even more powerful than vitamin C. A host of the colorful, vibrant antioxidants and phytochemicals that you find inside of the berries and herbs and different plants that we consume and take every day provide antioxidants, and they have lots of electrons to donate to the unpaired, unstable molecule. If this doesn't happen, this unstable molecule will then go and try to steal electrons from other healthy molecules, cells, tissues, and this is how, whenever you have a buildup of these unstable molecules around, that's where you get oxidative stress. So we daily need to try to fight free radicals in our body, or free radical production. And so these are some of the sources. You all know, we already talked about energy metabolism, smoking, radiation that we're exposed to, is also damaging our DNA. That's what the free radicals do once they start stealing electrons when they have unpaired electrons, that molecule, it directly damages your DNA. And also the air pollution, sunlight, of course, is also uh, providing lots of free radicals, the UV radiation. And what we see to the right of the screen, these are some of the many health concerns, the implications of excessive free radicals, our bodies not targeting the free radicals, AKA oxidative stress. So we have diabetes, neurodegenerative diseases, cataract development, rheumatoid arthritis, and of course, the number one concern with free radicals damaging our DNA is cancer. Because when you have unhealthy DNA replication, the end result is a cell that is no longer functioning properly and cancer can develop. So how do you know all of your antioxidants that you take every day, your blueberry, your pomegranate, you know, we mentioned your vitamin C, so many other things. Well, this is how you can measure free radicals. Urine, it's a urine test, and we are measuring F2 isoprostanes. And this is actually the gold standard for measuring free radicals. And they're so reactive, they're short-lived, so it's very difficult to measure, but these F2 isoprostanes are produced in a cascade that I'm sure many of you are familiar with. So many of you, I'm sure, take omega-3 fats, and we'll talk about omega-3 fatty acids in a moment. And many of you probably try to limit too many of the omega-6 fats that you find in dairy and meat and eggs. You may have heard of arachidonic acid, but those omega-6 fats provide arachidonic acid, which we see here. This arachidonic acid is typically, and I'll show the pathway to your left, typically uses an enzymatic pathway. And you see a couple of enzymes here, COX enzymes, COX-1 and COX-2. Those may sound familiar if you're thinking aspirin. Aspirin is an inhibitor of COX, or your ibuprofen. Many of your anti-inflammatory drugs target inflammation by inhibiting these enzymes. So the COX and LOX produce down this pathway different types of signaling molecules involved with the inflammation process. Now, what we see whenever you have excessive free radicals that have built up in the body, the foods that you eat, the fats that you consume, they do not go down the normal enzymatic pathway. They actually go down this pathway to produce this F2 isoprostanes. So it's just a product by the reaction of free radicals with arachidonic acid. And again, aging and age-related diseases reflect our inability to cope with oxidative stress over time. And the production of these isoprostanes is well documented to increase in direct proportion to our level of oxidative stress. And just to give you an interesting statistic, F2 isoprostanes may be elevated at the early stages of plaque development. So you do see many people testing this to look at whether or not they're developing plaque. But research has shown that people with high levels of F2 isoprostanes are 30 times more likely to develop heart disease. So for healthy aging, extending our longevity, we need to target free radical damage, and F2 isoprostanes is one way to do so. Moving on to the next mechanism of aging, excess sugar or glycation. We see this every day in the foods that we eat. If you love to grill, we have the 4th of July right around the corner. Who doesn't like a nice grilled anything. I like grilled portobello mushrooms, grilled onions, and grilled whatever you would like to have on the grill. What you do see is that that is an example of glycation reaction. The sugars in the foods combining non-enzymatically with the proteins in those foods, and they create a glycated protein. Now, what's important about the proteins in our body, they all have unique structures, and the structure determines the function of the protein. So whenever your proteins become glycated, then you start having proteins in the body that cannot function properly. So the reaction that occurs in the body, I gave you an example of outside of the body because we can eat some of these glycated proteins, but most of the glycation that you're dealing with is a result of excess sugar. Anytime there is sugar in the bloodstream, you will have a percentage of your proteins that will be glycated. If you are familiar with the hemoglobin A1C lab test, that is a test that's measuring glycated hemoglobin. And that's why it can give us an idea of your average blood sugar over a range of 2 to 3 months, because those are the hemoglobin molecules that have been exposed to sugar over time. Now, I thought about, that's one test that you should certainly consider as well. I didn't put it into my PowerPoint because I said, this is a smart group. They know about hemoglobin A1C. Let's think about something else that could be picking up on glucose spikes. And you will be surprised to see that two people can have the exact same hemoglobin A1C, but their glucose spikes after meal and overall exposure to glucose can be very different. So let's take a look. And just one note, we do know diabetics age prematurely and die about 10 years earlier than non-diabetics. And this process of glycation is one of the key reasons why we see individuals with diabetes dying sooner. So what we're looking at here is 1,5-anhydroglucitol. 1,5-anhydroglucitol is found in a constant state amount, or in a constant amount in your bloodstream. And it's also found in the foods that we eat. And you see here on the slide, I have an image of the glucose molecule, and this is 1,5-anhydroglucitol. So it's basically, it's like an analog of glucose found in the foods that we eat. Whenever we eat this, it's not metabolized. It's excreted in the urine in order to maintain a constant amount in the body. However, glucose spikes interrupts this entire process. Excess 1,5-AG, we'll just say that, is excreted, as well as the 1,5-AG that's needed to maintain the steady state of our body. So whenever glucose is around in excess, we start excreting this. So, hey, why not measure it? If we measure 1,5-anhydroglucitol in the bloodstream, and the level is less than what it normally should be, that tells us that we are having excess glucose spikes. So we see that food sources excrete it in the urine to maintain a steady plasma level. So let's take a look at, I kind of explained all of this, the slide is a little bit small, I apologize, but this is just showing food intake of this 1,5-AG, and we see that the amount that we usually take in as food is excreted, and we normally keep this tissue pool of 1,5-AG, goes into the bloodstream, goes to the kidneys, goes back to the bloodstream. So we keep a steady state here, is what we're seeing. Whenever you have glucose present, we see our tissue pool goes down because the excess glucose is causing us to excrete more than we should. This is the slide that I think really paints the picture. This is patient number 1, 7.4 A1C. Patient number 2, 7.4 A1C. But what we see here in the orange, patient 2 is having higher glucose spikes. And this is over, a couple of weeks here in time. So the date here is February 8th to February 15th. And you can see those post-meal glucose spikes, everything that you see in orange, that means that they are experiencing more glycation than they should. Versus this person, same A1C, but they're not having those glucose spikes. And what you see here is the glyco mark is what's reflecting the difference between these two individuals. And the glyco mark is the trademark name to measure the 1,5-AG, or that 1,5 glucitol. So that's another test for you to consider. Next mechanism of aging, methylation. Now, I'm sure many of you, if you've had any type of genetic testing, one of the big tests that people are looking into is to see if they're able to methylate properly. And most times we're familiar with the MTHFR SNP that some people have. And so this is just a quote from Craig Cooney. And in 1999, he was on to something. He stated, methylation is a major and fundamental determinant of health and sickness, or life and death. Like a car out of gas, life without methyl power comes to a screeching halt. So methylation is just a transfer of a methyl group, which is just one atom of carbon attached to 3 atoms of hydrogens. And it's the transfer of this methyl group from one molecule to another. And to date, every health conscious adult has a good reason to be familiar with it. Your body depends on methylation to detoxify carcinogens and other poisons, to repair damaged DNA, to form new cells, and to manufacture important anti-aging hormones. So the DNA within every cell of your body requires constant enzymatic reactions, and those enzymatic reactions, that's called the methylation. And this is needed to maintain and repair our DNA. And as we age, this process is crippled. And so the result is DNA damage. This can manifest as liver damage, cancer, brain cell degeneration, neurological health concerns, poor detoxification. And what we see here, this is the methylation cycle. And you can see different products in the cycle. Some of you are familiar with SAMe. We know that that's beneficial. When this cycle is not working properly, homocysteine begins to build up. In this cycle, you can see it's pictured as these cogs. And so if this wheel, if the red cog is not spinning properly, then it in turn affects the green cog, and so forth and so on. It becomes a chain reaction. So most times when we're looking at insufficient methylation, one of the key signs that people look for is excess homocysteine. We know that homocysteine is what builds up, it directly damages the endothelial cells. It's related to plaque formation, independent risk factor for developing plaque. And we can see oftentimes we are trying to give you things to help with this methylation cycle. Off from homocysteine, you see glutathione produced in this cycle. And some of the key factors that's needed in this cycle is B12, methyl B12. We also see vitamin B6 in this cycle. And what we're looking for here is methylfolate is needed in this cycle. And what you see that I've highlighted here, this is an enzyme that's needed to keep this cog going. Is the enzyme that activates folate into your active folate in the body. And some people may not have sufficient activity of this enzyme, and there's a way for you to test it. And so this is just going into some of the details that we went through already with the function and the benefit of the methylation reactions in the body. So it's more than just about homocysteine. Also needed for the production of CoQ10, carnitine, ATP. And so what I would suggest for you all to test is to see if you have a mutation. There are two common mutations to this enzyme or the gene that produces the enzymes that's needed to keep that methylation cycle going. And so this test will identify reduced enzyme function. And this is just showing some of the different mutations and the percentages of those mutations in the population. So you can see, white individuals, 25 to 45% mutation of those individuals will have a mutation in this particular gene that codes for this key methylizing enzyme. So this is something to keep on your radar. If you haven't yet tested to find out if you have this mutation, then you should do so. And this is just a sample report that I put in here to show whenever you receive your reporting, your genotype report, what you can look for, what the results look like. Next, the mitochondria. So we talked about the mitochondria, talked about how it's free radicals are produced whenever we are making energy. We know that ATP provides at least 95% of the cellular energy that powers all living functions. Cell death, also called apoptosis, is an essential part of life. As cells become old and broken, they are cleared away and destroyed. The mitochondria helps decide which of these cells are cleared away, these old dysfunctional cells that can potentially become cancerous. And these are just some of the other benefits of the mitochondria: storing calcium, generating heat. Your brown fat, that's the fat that we have when we're babies, where we're able to produce heat and just burn fat by just laying down and taking a nap. Hibernating bears have the brown fat, and we see that the mitochondria is involved with us having more of that brown fat. One key point about our mitochondria is that it contains its own DNA. That is a gift and a curse. It's a gift because we can regenerate new mitochondria. It's great that the mitochondria we once thought was symbiotic bacteria that has now evolved into we claim it as our own. But what you see here, this is our mitochondria, and one key component that's needed for the mitochondria to function is CoQ10. Now let's talk about the mitochondrial free radical theory of aging. According to a number of cell biologists, the number and functionality of the mitochondria specifically determine an individual's lifespan. Over 350 studies published in 2010 alone show how mitochondrial degradation leads to the onset of virtually every degenerative disease. Mitochondrial dysfunction can result in congestive heart failure, muscle weakness, fatigue, neurological disease. And we do know that there is a way to reverse this. So according to the mitochondrial theory of aging, damage to mitochondrial DNA from the massive amounts of free radicals that are produced during energy production is a root mechanism of aging. And the body produces CoQ10 to protect mitochondrial DNA. But we do know that our CoQ10 levels decline with age. And, a lot of this we can't necessarily feel. It may take decades. However, for this aging damage to become permanent, but we can reverse this with CoQ10. And what you see on this slide, I don't want to ignore it, but what you see are just some of the diseases. If you just Google mitochondrial dysfunction, there's now a mitochondrial dysfunction society, lots of research showing Parkinson's, Alzheimer's, chronic fatigue syndrome. These are all linked to mitochondrial dysfunction. So what we see here, CoQ10, when CoQ10 is low, mitochondrial dysfunction is high. And CoQ10, also known as ubiquinone or ubiquinol, we know that ubiquinol is the more absorbable antioxidant form of CoQ10. Both are beneficial, but this one is one that is particularly beneficial because it is an antioxidant. It's called, CoQ10 is called a coenzyme or coenzyme Q10 because of its unique ability to participate in chemical reactions and remain at a steady state in your cells. And so in the electron or in the energy production in your mitochondria, CoQ10 is involved with scavenging free radicals. It's an electron donor, and this is what kind of keeps our body going. Now, just some of the things that can cause CoQ10 to deplete, or for us, that some of the causes that would cause us not to have sufficient CoQ10. One in particular is our diets. Organ meat and seafood. These are the dietary sources of CoQ10. And many of us probably aren't having liver regularly or brain, depending on where you live, or there are some cultural influences on this, but most people, that's not a staple in our diets. And what we typically do consume is poorly absorbed. But other things that we know deplete CoQ10, many drugs, statin medications. If you are on a statin medication, it is directly inhibiting your natural production of CoQ10 in your liver. That is how the statins work in the pathway of how it's blocking cholesterol from being produced. It is also blocking CoQ10. Antidepressants, beta blockers. If you have high blood pressure, you're on the beta blocker, you are depleting your CoQ10. And there are also some anti-diabetic medications that also deplete CoQ10. So you want to test CoQ10. And it's a simple blood test. And here are a couple of guidelines. There are two strategies. You can, number one, get a baseline for your CoQ10, and then repeat after 4 weeks of taking CoQ10. Or you can start your CoQ10 right away, which I typically suggest. Take CoQ10 for 4 weeks, and then test your levels to assess if your CoQ10 is within healthy longevity optimal ranges. So what are those optimal ranges? You want your plasma CoQ10 to be at least 7 micrograms per milliliter. In order to get to that level, you need to take about 1 to 300 milligrams of ubiquinol CoQ10 per day. Now, if you have, let's say you are on a statin medication, you may need more CoQ10 or more ubiquinol because your statin medication is depleting your CoQ10. So it's likely that you may need more, but the key point here is to test to make sure you're getting into your optimal anti-aging ranges. And our last mechanism of aging is chronic inflammation. Now we know acute inflammation, it's normal. It's how our body heals. If you stump your toe, if you cut yourself, you're gonna feel the heat. You'll see the swelling, you'll feel the pain. There will be redness. And, you know, under normal conditions, all those immune cells that's in there cleaning up your inflammation, after performing their action at the inflamed site, they go away. They go away, your inflammation subsides, and you regain your function. But chronic inflammation, what we see here in red, this is immunopathological changes such as infiltration of inflammatory cells, overexpression of pro-inflammatory cells and genes, dysregulation of cell signaling, and loss of barrier function. Chronic inflammation is connected to a host. Well, you know, at Life Extension, we actually state chronic inflammation is the common denominator for all age-related chronic diseases. Neurodegenerative diseases, diabetes, cancer, obesity, a host of inflammatory diseases. We know that you're dealing with chronic inflammation. This is more of your systemic low-level inflammation. It's inflammation that you can't necessarily feel, but it's there, and it's causing damage to your tissues, to your cells, causing fibrosis in your body, and it is a key mechanism of aging. So many of the things that you're doing is targeting inflammation. But one of the best ways, or one of the best nutrients to target inflammation, are omega-3 fatty acids. Now, this next slide is really busy. Just brace yourself, but I promise the key, I will kind of hone you in to exactly what's important for you to view on this slide. So what we see here are omega-3s and inflammation. On this left side of the slide, you see a more pro-inflammatory state. And on the right side, you see more of a healthy anti-inflammatory state. Now, what is the difference between these two sides? What we see here is a person that is consuming an excessive amounts of omega-6 fats in their diet. We talk about the SAD diet, or the standard American diet, full of omega-6 fats or arachidonic acid. That arachidonic acid, whenever we eat those fats, any type of fat that you eat, those fats get incorporated into our cell membrane. So that's what you see. These are two cells, and this person ate lots of fried chicken, McDonald's, Taco Bell, all the places that I'm sure none of you would ever eat, right? But you would be surprised that there are lots of omega-6 if you eat lots of eggs. You know, you're thinking that you're being healthy, you're eating your eggs in the morning and maybe some avocado toast, you still may be getting omega-6 fats. And those fats incorporate into your cell membrane. And whenever your body needs to fight, or there's an injury, we need inflammation because we know it's a normal healing process. It pulls those fats from the cell membrane. And remember, we showed earlier that those COX enzymes, this is the LOX enzyme, and you have these inflammatory mediators that are produced. And some of these, the ones on this side that's produced from arachidonic acid, are powerful. You have prostaglandins, thromboxanes, leukotrienes, and these tend to be pro-inflammatory. Now, on the right, let's say this person is eating lots of good fatty fish, tuna, salmon, sardines, anchovy, chia seeds. They're sprinkling their ground flaxseed on their salads. They are eating lots of walnuts. They are going to have more omega-3 fats in their diet, hence more omega-3s in their cell membrane. And whenever the body needs an inflammatory response, it will pull those fats, your good EPA, DHA, from your cell membrane to make inflammatory mediators. But the main difference between the mediators that's made on the EPA, DHA side versus the arachidonic acid side is that these have less of a strong inflammatory response in the body. They can get the job done, but it's not so strong where it's going to cause chronic inflammation. And unfortunately, just in general, you would be surprised when we compare omega-6 to omega-3 in individuals. Ideally, in an ideal world, you would like there to be about a 1-to-1 ratio. Life Extension knows that it's probably, everyone's not going to be that perfect. You may find people that's at a 2-to-1 ratio. And then others, we feel like it's acceptable to have about a 4-to-1 ratio if we're kind of comparing your omega-6 to your omega-3s. The typical person consuming the SAD American diet, or just in general, most people, their ratio is about 23-to-1, which explains why in our society we're so inflamed. We're dealing with chronic inflammation, and we're dealing with an overabundance of chronic age-related diseases. So what should I suggest for you today? My suggestion is for you to test your omega-3s. Know where you are. If you're taking omega-3 fats, great. If you're not, that's even a better reason to test. I went on a quest at one point to say, I don't need to take fish oil. My diet's great. I'm eating my fish. I'm eating everything right. I'm limiting my omega-6. I don't need to take an omega-3 supplement. And guess what? Even when you think you're doing right, whenever you test your omega-3s, you will be surprised. So this is your omega-3 index. And this is just showing you your typical reference range. This is a sample of 99% of US adults will be in the range of about 2.9 to 12.9. And you can see this person, this is their index. This is where I want all of you in the room to be. This person's index is 13.01. We need you to be an overachiever. That is important, because when you are overachieving here, that means that you are combating inflammation in your body. And this is just another example of a report here. You can see you can measure omega-6 to omega-3 fats. This person is a 2.1-to-1. Who in this room thinks that their omega-6 to 3 is this good? Yes. If you're not sure, that means that you need to test. And this is looking at arachidonic acid to EPA, another ratio for your omega-3s. And these are just some of the strategies that you should consider. So just for the summary, and that was our last slide, we want to target oxidative stress. We want to inhibit glycation in our body. We want to support healthy methylation. We want to support mitochondrial function, and we want to make sure that we are combating excess inflammation in the body. And these are 5 lab tests that you should consider to make sure you're on the right track. Super. I know you are on the right track.
Dr. Gossard, thank you so much. Can you hear me?
You're welcome. I can, yes.
This is Jim Stroh.
Hi, Jim.
Yeah, and my partner here, Bernardine, is sitting with the directors of People Unlimited. And I think, have I seen you at RadFest? You look familiar. You been at RadFest?
Excuse me. I have not had the pleasure of going to RadFest, but one of these days, we sent at least 2 to 3 people from Life Extension, but I've been going to some of the other conferences, maybe A4M. I've been there several times.
Okay, but you need to be at RadFest. You haven't been to an event until you come to RadFest. So. Yeah, but no, you did such a great job.
That's what I hear. Thank you.
Yeah, thank you for doing such a great presentation. It's really informative. Yeah. So I think we have
Thank
some
you.
questions then, if you have some time.
Yes, yes.
Thank you. Okay.
Okay, so we have a issue with the camera, so Dr. Gossard can't actually see you, so but she can hear you. So go ahead.
Hello, this is Lori. Listen, I really liked your presentation. I don't usually tell people that, and you are my favorite. Yeah, I do have favorites. You were so scientific, but very simple, but not too simple for us, and that's quite an achievement.
Thank you.
So questions for my favorite. On the urine test, I assume that it's a 24-hour urine test, but is that correct? Is it a 24-hour urine test?
No,
For
so
urinary F2 improstains?
no, no, it is not a 24-hour urine test. It's just a one sample, and believe it, and it's actually able to kind of identify the glucose spikes in a range of about 4 weeks, 3 to 4 weeks. So that's where you kind of start seeing within that time frame the F2 isoprostanes slowly declining.
Okay, good. Thank you. And also for my baseline CoQ10, since we are overachievers, is there a risk of being too high, like way above the range? Is there a problem with that?
So I've looked at lots of CoQ10 labs, and you know, you all are overachievers. A lot of our life extension, we call them the life extenders. They are also overachievers, and we have not seen anyone that's over the 7 micrograms. But let's see, could you have too much CoQ10? I would have to say no. I wouldn't imagine. I've never seen it in any of our laboratory assessments.
He says thank you.
You're welcome.
Yeah, thank you, Crystal. I liked your upbeat attitude. I could tell that you really enjoy what you're talking about. And you
I
know
do.
Yeah, fantastic. I'm in touch with Dr. Jack Cruz, who talks, well, he's got a gang of us that he calls mitochondriacs. So one of the main things that he's bringing out now is the effects of artificial light, especially the LEDs, and especially the fact that we get up in the morning and the first thing we do is look at our devices, and we're depriving ourselves of the natural sunlight, especially early morning light, to kind of like set our circadian rhythms. And he says this is the primary food. We're just starving ourselves. And so he's getting a lot of good feedback from many people about when they correct this with the yellow glasses and so forth.
Yes.
Would you like to weigh in on that?
Oh, you are absolutely on to kind of a new area of science. I did a talk on circadian rhythms a little while back, and just the research looking into your clocks. So you have a central clock in your brain, and then you have these circadian clocks all throughout the body in every cell. Now, whenever we get that good sunlight, the natural light, especially first thing in the morning, I encourage you, get outside, open your windows. That is the first thing that you should be doing. That's helping to put us in sync with our environment and to sync our clocks, our central clock. And that central clock then communicates to the peripheral clocks in our bodies. And those peripheral clocks, specifically, they're clock genes. And those clock genes need to be activated from the central clock in the brain. And those clock genes control everything. Those genes are controlling our metabolism, hormone production, neurotransmitter production. So it's the excessive artificial light, the blue light, going to bed. A lot of people go to bed and they have the last thing they're looking at is their smartphone before bed, getting a little FaceTime or Facebook or Instagram time. We have the sleeping with the TV on, that artificial light. All of that is suppressing our production of melatonin, which is also needed for our clocks to run. And we do find a host of diseases associated with disrupted circadian rhythms. And so I encourage you to continue to incorporate your strategies. Good job, all of you in the room. That's the most important thing, your blue blocking glasses. You can also buy, on my phone, I have it where at 6 or 7 p.m., it's on a timer, it automatically switches to night mode. And so it kind of puts an orange hue on your phone to block out the blue light. The same thing happens on my computer. It's automatically set to switch to night mode. My husband, he's really into this as well. We changed out the light bulbs in our bedrooms, and now we have like an orangish red hue light bulb. And my husband, he puts his glasses on, and as you can tell, I'm pretty upbeat. So he's just like, okay, Crystal, here's your glasses, put these on, because it's time to really start getting into nighttime mode, and that's very important.
Thank you, Dr.
You're
Chris.
welcome.
Fascinating. I love this subject. I want to ask something that always bothered me. I know I need CoQ10, but I know that I need the omega-2. So I eat those grains, seafood, liver, whatever that produce the fatty acids. Okay, so I know also that the CoQ10 is fitted too. So do I need to take also CoQ10 when I did all this diet, like it's a lifestyle of food? And what do you prefer, to take a supplement or to be full of CoQ10 from the food? That's my question.
Okay, excellent question. I wish that food would be all that you needed for CoQ10, but it's not. And it's unfortunate, but even the CoQ10 that you would find in the organ meat is poorly absorbed. So you, I would say if you had to choose out of CoQ10 and omega-3s, which one you would want to focus on getting from your food and which one you would want to supplement with, 100% supplement with CoQ10. The omega-3s, if you are on top of it, you can get your omega-3s from your food. From if you have, fatty fish 2-3 times a week, and have the flax and the chia and the walnuts, 100% you can get your omega-3s in your food. So if you wanted to choose between the two, that would be my suggestion.
But see, you could also test that. You could test your CoQ10 levels. That's the beauty of your approach, right? So
yes,
you could actually really find out what the status is. So
Yeah, I agree with you on the supplementation. My concern with the omega-3s is that, and eating the fish, fish is so polluted nowadays. Do you think? Yeah, and some of these good supplements have been tested for, mercury and so forth, and are, relatively pure. So that's why I tend to do more supplementation than eat a lot of fish. Would you say that's pretty true?
That's 100% true. Your fish, there's lots of lead and PCBs that you'll find, and lots of pollutants in the fish. And so that's one of the downfalls, because nowadays, we as nutritionists, we don't recommend the fish more than 2 or 3 times a week. And if you're choosing the fish route, try the smaller fish, because they're less likely to have the contaminants, because we know that the pollutants, they bioaccumulate So the larger the fish, they're eating the smaller fish. And so they will have more of the heavy metals and the toxins. So if you're looking for fish, do your anchovies or your
right.
sardines if you decide to go that route. Yes.
Right.
Very good.
There's some of the cleanest fishes out of Alaska too. I don't know if that's totally true still, but
It is true.
yeah. Okay.
It is wild Alaskan, and the season just started. So you should be able to find, and if it's not in season, you definitely need to supplement outside of season, because there's a lot of food fraud going on now. And many of the restaurants that's saying that it's wild Alaskan, if it's out of season, probably 70-80% of the time you're getting scammed. Not the real thing.
Okay, so I'm a nurse, and one of my patients I give for the fish oil, his mom wants him to have calamari oil. Is there a difference between the calamari fish oil or fish oil pills? Just like, which one's better? Is there a big difference between the calamari?
Now, some people like the calamari because it's, if they are kind of sensitive and they repeat their fish oil, they kind of have the fishy burps and things like that. It's usually not as fishy. However, most of the fish oils, if they are pure, molecularly distilled, you typically won't have any concerns with them being have that fishy kind of burping and things like that. Some people also like the calamari oil because they may be getting, a lot of time those will give you some of the other fats and things like that, fatty acids other than EPA and DHA, but there's no big difference. What's most important for you to look for is the amount of EPA and the amount of DHA, regardless to whether you choose krill or salmon oil or, fish oils from other sources. You want at least 1,000 milligrams of DHA and at least about 1,500 milligrams of EPA. That's what's most important. You want to look for those doses because a lot of even your vegetarian options, we know that now you can get your omega-3s from algae, and it's a great vegetarian source of omega-3s, but usually you may not be receiving those higher amounts of the DHA, and you would have to take multiple soft gels in order to get up to the higher amount. So that's what's most important.
Okay, so does Life Extension have a offer on, can you get, I know sometimes they do packages or on these panels. You have to get these tests separately, or is there a panel that you can just get and get all these on it?
Oh, there is no panel with all of these, unfortunately. But, offline, I can talk to our laboratory manager and our director of laboratory services to see what we can do in regard to offering something for your group.
Oh, that'd be great. Okay.
Yeah. I'm sorry we didn't plan it sooner, but yes, we can do something.
That'd be great. I think most of us would be interested in doing that test.
Yes.
So, okay, good.
Okay, I think that's it. So thank you so much, Dr. Gossard. That was outstanding presentation.
You're so very welcome. You all are such a lively group. I love it. You energize me.
Thank you so much.
You're welcome.